Okay. Thank you, Jordan. Thanks very much. And I'm going to talk about transgenerational transmission of the effects of nicotine exposure with some more data on the effects of saccharin and aspartame as well. So let me just see here. Our interest in prenatal nicotine exposure is also motivated by the association between prenatal nicotine exposure and ADHD. So some years ago, we published a paper that showed that if women smoked cigarettes when they were pregnant, the risk for ADHD in their offspring increased significantly, almost doubled. In addition, ADHD due to prenatal nicotine exposure is symptomatically indistinguishable from ADHD due to other causes, which means if you're working with animal models, an animal model of prenatal nicotine exposure has significant construct validity as a model for ADHD. With that background, we developed a prenatal nicotine exposure mouse model. As you can see here, we exposed C57 black female mice to nicotine in drinking water, beginning 3 weeks before conception, continuing all the way until birth. At the time, this was 2012, we thought that mice will not drink water that contained nicotine because nicotine has a bitter taste. So we added saccharin as a sweetener, which meant that we had two control groups, mice that only received saccharin, mice that received plain drinking water. So we had these three groups of mice exposed 3 weeks before conception, during pregnancy until birth. Our goal was to characterize behavioral phenotypes, behaviors that would be consistent with ADHD symptom domains, hyperactivity, attention deficit, impulsivity, and two comorbid conditions, working memory deficit and cognitive inflexibility. So we have basically cognitive and one motor behavior that we analyzed. What we found, we began with spontaneous locomotor activity. So here I'm showing you data from spontaneous locomotor activity in these mice. Activity was recorded continuously over approximately 17 hours, predominantly during the lights off period of the light-dark cycle. Mice are nocturnal, so they are more active during this lights off period, natural period of activity. You see two dark lines here. They both belong to the control groups, water and saccharin only exposure. You see an increase in activity during the lights off period. As I said, this is a natural phenomenon for nocturnal animals. What about mice that were prenatally exposed to nicotine? You see a significant increase in spontaneous locomotor activity, or these mice are hyperactive. These are males and these are females. So both male and female mice showed significant hyperactivity as a result of prenatal nicotine exposure. What about the cognitive behaviors? So as I said before, we measured attention, motor impulsivity, and working memory. Male offspring, or male mice prenatally exposed to nicotine, had attention deficit, impulsivity, as well as working memory deficit. Female offspring only had attention deficit. So here we begin to see the effect of sex on the manifestation of these behavioral changes. At that time, transgenerational transmission of environment-induced phenotypes was becoming more and more recognized. So as you heard a number of times, smoking was highly prevalent in the 50s. So there's reason to examine whether prenatal nicotine-induced phenotypes show transgenerational transmission. So with that background, we embarked on a transgenerational transmission study. And here, as you can see, relatively simple to do these experiments in mouse models and in human studies, as you heard. So this is the F0 generation, which is the female mice exposed to nicotine before pregnancy and during pregnancy. The offspring, F1, which I already showed you, were hyperactive, had attention deficit, working memory deficit, and impulsivity. We took these F1 mice and crossed them with drug-naive partners to produce the F2 generation. So if you go back to the human scenario, this would be the grandmother, mother, and this will be the grandchildren. We found that the F2 mice, male and female, were significantly more active, or they showed hyperactivity. We also produced an F3 generation. The F3 mice also were hyperactive. So these data showed that smoking or prenatal nicotine exposure produced hyperactivity in the F1, F2, and F3 generations. What about so this is transmission of a behavioral phenotype. What about the putative or potential neurobiological mechanisms associated with these behavioral phenotypes? Are those neurobiological changes also transmitted transgenerationally? So to give you one example of how we addressed this issue, in the F1 generation, we knew that treatment with the stimulant drug methylphenidate or Ritalin significantly ameliorated the hyperactivity. A saline-administered F1 mouse is hyperactive. Administration of methylphenidate mitigates that hyperactivity. We found the same types of data in the F2 and F3 generation as well. In males and females, administration of methylphenidate significantly reduced spontaneous locomotor activity. We had shown before that methylphenidate administration increases dopamine in cognitive centers of the brain, particularly the frontal cortex. So these data show that not only are the behavioral manifestations transgenerationally transmitted, but potentially the neurobiological mechanisms, for example, neurotransmitter signaling, are also transgenerationally transmitted. The changes in these mechanisms are transmitted transgenerationally. At this point, we became aware, like I think most people already knew, that more men smoke cigarettes than women. So what may be the potential of transgenerational transmission of nicotine-induced phenotypes in male mice or in fathers? You already have heard studies from human populations. So we also went back to our databases in collaboration with our child psychiatry colleagues, and we found that if smoking by the father at the time of conception increased the risk for ADHD in the offspring. So that gave us a framework to develop our animal model. So we went back to our mouse model, and here we exposed male mice to nicotine for approximately 12 weeks. Control mice received plain drinking water. By this time, we realized C57 black mice, at least, drink water containing nicotine. They do not need saccharin sweetener. So that saved us a lot of work and resources. We could only use a single control group that received only plain drinking water. So we began by analyzing behavior in the nicotine-exposed male mouse, or the founder, the F0 generation. We did the same analyses: locomotor activity, attention, impulsivity, and working memory. We did not find any significant difference in these behaviors in the nicotine-exposed and plain water-exposed mice. So the founder generation did not have behavioral changes. We produced the F1 generation by crossing the nicotine-exposed male with drug-naive female, and we found that in the F1 generation, male mice were hyperactive and showed attention deficit. Female mice were hyperactive, but did not show any of the other behavioral changes. So 2 points to make. Even in the paternal exposure paradigm, there is an effect of sex of the offspring in the manifestation of these behaviors. And second is that these behaviors are different from the prenatal nicotine exposure model, where we also had found impulsivity and working memory deficit in the male offspring. We continue to examine another behavior in these F1 offspring, that is cognitive flexibility. The mouse counterpart is reversal learning. This is the ability to shift between different concepts or simultaneously tackle multiple mental tasks. This, for a mouse, and I think for us as well, is somewhat more challenging behavioral task. It not only requires intact working memory and attention, but also requires coordination of previously learned responses with working memory and attention. And this is a core symptom of autism and also a key symptom in other neuropsychiatric conditions, ADHD, OCD, and anorexia. So we generated F1 mice and also this time went to F2. So we had the children and the grandchildren of the nicotine-exposed male mouse. We found that both F1 and F2 mice showed significant cognitive inflexibility. In summary, what our paternal nicotine exposure mouse model shows is that although the exposed males in the F0 generation did not have a behavioral phenotype among the phenotypes we had measured, F1 and F2 mice had behavioral changes. Why am I showing this rather large table is because it makes 2 points. One, the phenotypes show attenuation from F1 to F2. F1 mice were hyperactive, had attention deficit and cognitive inflexibility, but in the F2 generation, we only saw cognitive inflexibility. And the second point is that sex of the offspring and the founder had a significant effect on the manifestation of these behavioral phenotypes. Sex of the offspring is clear from this table. Males and females have different phenotypes. But in the case of cognitive inflexibility, in the F2 generation, the deficit was only in male mice, only if they were derived from a male founder, F1. If they were from a female founder, we did not see that difference. So in this paternal nicotine exposure mouse model, we saw that the phenotypes attenuate and sex of the founder as well as the offspring had a significant effect on the outcome. Going beyond behavior, what about the neurobiological mechanisms? Again, something we can do in a mouse model, but not in the human studies. We focused on dopamine receptor signaling because if one looked for a common denominator in terms of neurotransmitter signaling for all the behaviors we talked about, cognitive and motor, dopamine would be at the top of the list, and I hope you would agree. So we examined dopamine tissue content and dopamine receptor messenger RNA expression in our paternal nicotine exposure mouse model in F1 and F2 generations. These are ongoing studies, so we don't have a lot of data, but what we found was that in the striatum, which is the basal ganglia nucleus involved in attention, working memory, hyperactivity, etc., there was a significant reduction in dopamine content in male mice. With respect to dopamine receptor mRNA expression in male mice in the striatum, D1, D2, and D4 receptor expression was reduced, but in the female, D5 receptor mRNA expression was increased. F2 generation also showed differences between males and females. So not only were the behavioral phenotypes dependent on the sex of the offspring, even the molecular phenotypes and neurochemical phenotypes were influenced by sex. And the next experiment was the mechanisms of heritability. Of course, we looked at epigenetic changes. And this is another reason we actually moved to a paternal nicotine exposure paradigm, because collecting germ cells from male mice is a lot easier than from female mice. So we collected spermatozoa from the epididymis of F0 generation, that is, male mice exposed directly to nicotine for 12 weeks. And we found a significant increase in global DNA methylation in the sperm and significant decrease in dopamine D2 receptor promoter region methylation. We looked at the promoter regions of all dopamine receptors. We found significant changes only in D2 in the spermatozoa. But still, dopamine receptor gene promoter region methylation was significantly altered by nicotine exposure. We have not yet done a methylome analysis of every gene and every promoter region, but we focused on dopamine and we found changes in the spermatozoal DNA methylation. So in the nicotine exposure model, we found behavioral phenotypes, neurochemical and molecular phenotypes in the brain, and then also DNA methylation changes in the sperm. So what about other environmental exposures? Is this a nicotine-only phenomenon in mice, or other exposures also produce similar results? So we began with saccharin exposure because we had in the past used saccharin for prenatal studies, but here we exposed male mice to saccharin and examined the behavioral changes in the exposed generation, so F0. The mice were impulsive. When we went to the F1 generation, meaning the fathers were exposed to saccharin, F1 males were hyperactive and impulsive. We did not see any significant change in the female F1 mice. Again, sex differences here too. Then we said, what about co-exposure to saccharin and nicotine? So we exposed male mice to saccharin and nicotine, and we analyzed behaviors in the exposed generation first. The male mice were impulsive when they were exposed to both saccharin and nicotine. F1 generation, males were hyperactive, impulsive, and had working memory deficit. Female mice in the F1 generation did not show significant changes in behavioral parameters. What might be a reason for doing the nicotine only, saccharin only, and then co-exposure? Well, I think it told us something interesting. First, it told us that nicotine plus saccharin exposure is not merely an additive effect of each exposure. For example, you will see from this table, nicotine produced hyperactivity in males and females. Saccharin produced hyperactivity in males, nicotine plus saccharin only in males. But more interestingly, attention deficit produced by nicotine in the F1 mice was not seen when nicotine plus saccharin were administered. The impulsivity produced by saccharin exposure, not by nicotine exposure, was present in the co-exposure. There was a working memory deficit in the co-exposure not seen in either exposure alone. So the point that I wanted to make is that knowing the effects of a particular environmental insult or two environmental insults by themselves does not necessarily tell us the outcome of co-exposures. Co-exposures are an emerging phenomenon, not simply the addition of each of the two. This might be of interest when people talk about environmental exposures, like the multiple components in cigarette smoke or air pollution, et cetera, et cetera. DNA methylation also showed basically the same thing. The co-exposure is not merely addition of nicotine and saccharin. Finally, in the remaining time, I want to talk about exposure to aspartame, another low-calorie sweetener. It's not that we're just going down a list of low-calorie sweeteners. The reason for aspartame was that aspartame is considered safe for human consumption. Generally speaking, we focus on adverse stimuli, nicotine, cocaine, alcohol, high-fat diet. But what about something as mundane as aspartame, considered to be safe? So we exposed male mice to aspartame, and we produced the F1 generation. There are some differences in the phenotypes that we tested and we observed in aspartame exposure compared to nicotine and saccharin. We found that aspartame exposure of the male mice in the exposed generation produced significant anxiety-like behavior. This was seen also in the F1 males and females. We did not see attention deficit. We saw working memory deficit in the F1 generation, but not in the F0 generation. There were some studies suggesting that aspartame exposure produced depression in clinical studies. We did not see that in our model. So here, aspartame exposure did not produce the same phenotypes as nicotine or saccharin. An interesting thing here was the working memory deficit arose in the F1 generation, even though the F0 mice did not show that. Something else that we are able to do here, because our phenotypes could be measured repeatedly in the same animals, is we found that 12 weeks of exposure was needed to produce phenotypes in the F1 generation. 8 weeks of exposure, although it produced anxiety phenotype in the F0, did not contribute to transgenerational transmission. So the duration of exposure also was a factor in successful transgenerational transmission. So in summary, what I think, or I hope you agree, our data show is that heritable behavioral phenotypes are influenced by the sex of the founder as well as the offspring. Phenotypes tend to attenuate with successive generations because the insult, the environmental insult, is transient in our model. Only the founding population was, or founding generation was exposed to the environmental insult. And the transmission is not only limited to behavioral phenotypes, but also to neurotransmitter and molecular phenotypes. The phenotype of the founder does not consistently predict the behavioral phenotype in the descendants. And there is a window of vulnerability, both for expression of the somatic phenotype, or in the founder population, the behavioral phenotype, as well as the heritable phenotype. It appears that, at least in the aspartame exposure, founder phenotype emerges before heritable phenotype, perhaps suggesting the protection that germ cells have in mammalian, in mammals. And finally, I want to leave you with an ongoing study. So we talked about behaviors, neurotransmitters, and molecular changes in the exposed generation as well as the descendants. What about the structure of the brain? So I think, at least in neuroscience, if there is transgenerational transmission of structural changes, I think we would, it will help advance this field even more than merely behavioral or neurochemical changes. So what we found is that in our prenatal nicotine exposure mouse model, using diffusion tensor imaging, there were significant structural organization changes in the medial prefrontal cortex, dorsal striatum, and 2 areas of the cerebellum. And we analyzed the entire brain, and these were the only 4 regions that showed significant changes. This is preliminary data, but the interesting thing about this is in untreated ADHD, these same brain regions show both functional and structural alterations. Therefore, this may be a relevant phenotype to study in addition to behavior and molecular changes. So with that, I want to conclude and thank you all for listening and acknowledge the contributions of numerous people in my own lab. Deirdre McCarthy and Sarah Jones presented their findings a couple of weeks ago. Lin Zhang has been active in the prenatal nicotine exposure mouse model. Also wanted to thank my collaborators at Mass General Hospital and University of Florida. David and Brad did the neuroimaging studies, and Joe for one of the first funding that we received for this work, and the NIH and FSU. Thank you.